Students, so today we are discussing about ninth class biology and first chapter. Cell is the structural and functional unit. Cell is the basic unit of life. So in that activity 1 we are going to discuss. So activity 1 is main to know about plasma membrane structure and its function, and how can we see the plasma membrane. So to see the plasma membrane, mainly we can take a best leaf, that is rheoleaf. So rheoleaf is best suitable to observe the plasma membrane to conduct the experiment. Now first of all you have to take the rheoleaf, and so the complete name of rheoleaf, Tradescantia spathica. Tradescantia spathica, the complete name. And so we can conduct this experiment easily. You have to keep in your mind, Tradescantia spathica is the complete name of complete rheoleaf. So you can you have to take this leaf and tear randomly, and whatever thin white layer is comes and keep onto the slide, and you have to leave the water 2 drops on the slide and cover the cover slip and kept under the microscope, and we can see this diagram after keeping under the microscope, that rheoleaf after tearing and keeping the white part, that whatever the part which is comes when we tear the random. And we can see here cell walls, we can properly we are observing, cytoplasm we are observing, nucleus also we are observing in this. So we can see this structure, and when you keep the rheoleaf in the microscope. So when we add the salt to the same cells, same part of the rheoleaf, what happens? Keep under the 5 to 10 minutes. All the plasma membrane, so you know that plasma membrane, so imagine this is the cell. So plasma membrane is present below the cell wall. This plasma membrane, what happens? It will be shrunken when we add the salt. So that after that what happens, when we add the salt to the cell, the plasma membrane will be shrink like this. It will happen here. The plasma membrane is normal, but when we add salt, the plasma membrane is shrunken. So in this way, before adding salt we can see this one like this plasma membrane. After adding salt we can see like this. So this is the before adding the salt, after adding the salt. So these pictures you can observe and compare. Let us know that here we can see plasma membrane clearly and without shrinkage, but here we can see the shrinkage of plasma membrane. Like when we remove balloon will be shrink like that. Here in picture B, the plasma membrane will be shrink after keeping the salt, after adding the salt water. So that salt water makes to shrinkage the plasma membrane. So by this we conclude that here the important process, osmosis, is occurring due to adding the salt.